Now we are going to find out what happens to that blastocyst and how it implants. We saw it going down the fallopian tube into the uterus, and then it'll stick to the wall, to the lining of the uterus. So that's where we want to pick up right now. And here at the top it says week 2 begins with implantation. And of course that's when the preembryo implants into the uterus. The embryo attaches itself to the wall of the mother's uterus. So we're going to zoom in on this drawing first. It says by the end of week 1, the embryonic cells have used most of the energy that was in the original ovum egg cell. Remember, the egg cell had lots and lots of little fats and sugars and good things to eat all stored up. And by now that energy is starting to run out, so they're going to need to find another source of energy, which of course is going to come from the mother. So we're going to make this into our little blastocyst. And this is the last time you're going to have to draw these cells. From now on, I'm going to draw them for you, and eventually we won't draw them at all. And then we want to draw that inner cell mass. And I like to put a little kind of line around it to make sure they stand out. And then if you'd like to, we can even do that shading trick where we could make this look hollow, because of course this is a cutaway. If you were really looking at it from the outside, it would just look like a ball of cells. You wouldn't really see the inner cell mass. So this is our cutaway view. So you can see what's going on the inside. And you can always use your finger to help kind of shade a little bit. Go around the top a little, and make it just a little bit darker around here. And then you can always make some more bumps like this so you can see that the texture is going to the inside. There's lots on the far wall. Okay, so there's our blastocyst, and we need to make it touching the lining of the uterus. So the lining is going to be over here. So make a line right about something like this. There it is, touching. And as soon as this happens, some cells start growing out like this. And these cells look like they have fingers. Now we'll see this at other places in the body too. We'll see cells that look like they have fingers. And the name for the fingers is villi. So we could point to these fingers, call them villi, V-I-L-L-I. And that means fingers. It means little fingers. Now on this side here, we're going to draw some red capillaries. Little tiny blood vessels coming over, and of course these would be the mothers. And they'll come over and almost touch. Now this is what these little cells are reaching for. They need to tap into the energy here. Now blood is never going to mix. When the baby eventually has blood, it's not going to mix with the mothers. They'll always stay separate. because this is a separate individual, right? You might not even have the same blood type, so you don't want to mix blood. But they're going to get so close that little bits of sugar and oxygen and things can leak back and forth. So let's make these red so that they look like little blood vessels. Now, some also would be blue, you know. You can't forget that in here, you don't have to do this, but like this one would bend around, right? It would come, it's a continuous loop, and it would go back out like this, and there'd be, yeah, maybe this will come over like this. There would be return vessels as well. So we get kind of lazy when we draw. We just make these little red things coming over, but to be correct, they would loop over and then they would go back. But even if we don't make the blue loops, we get the idea. The most important idea is to know that there's little capillaries over here. And that's what these little villi are reaching for. So I think I'll make some colored background. I'm going to choose, this is kind of like, I don't know what it is, this is called peach. You can use, don't use yellow though, we're going to use yellow for something else. If you want to make a little kind of off-white or peach or pink would be okay. Something very, very light, very, very light tan. Or you can leave it if you want to. I just thought it would be nice to make it obvious that this was all part of one tissue here. So what I'm coloring is the lining around the uterus called the endometrium. And in humans, every month, the uterus grows a new endometrium. Okay, so this is the endometrium, which is the lining of uterus. So there's more tissue back here, and these blood vessels would go back and connect and go to bigger blood vessels. But this very outer layer of the uterus will grow little blood vessels. Just in a matter of days, these can grow. Just for a week or two, it grows all these tiny blood vessels. And then if this doesn't happen, there's no embryo that attaches, then this will all disintegrate and shed out. And that's what we call a menstrual period, is this, of course, lining draining out. And then the next month, it'll grow a new lining and all new blood vessels will grow. And if we want to be fancy, you can even add, there's some other things in here. There's some glands that grow too. They just look like there's a little bulb area on the end that is where the products are made. The cells are making little proteins and sugars and things. And then they go out like this. So if you want to be fancy and correct, I'll add one behind here. There we go. Okay, so these are glands, and they would make, I read that they make mainly proteins, some sugars and things. Basically, they would nourish this ball of cells. They would help encourage it to grow and nourish it while it was trying to establish a connection here. It's real hard to find good information on these things. They just call them uterine glands and leave it at that. So, but what I read is that their job is to help nourish the little ball of cells. Okay, so let's go over here and find out what happens next. Zoom out just a little here. So This is by day 8 or 9. Alright, so you see that I've drawn the outside ring of cells now. We're kind of zooming out. You can see we've reduced the size so that this is probably maybe a little more realistic. These are probably a little bit large, but you're still going to have to draw that inner cell mass. So actually this is probably the last time you'll have to draw this, because the next thing we're going to do is zoom in on just this inner cell mass and find out what happens to it. Okay, and for our line, we're not going to put that line down here. What's happened is that those little cells here, you can make some of them reaching out. We're going to draw these again. They've done something else. They have released enzymes, scissor enzymes, digestive enzymes, that have kind of eaten away at these tissues. They may have actually dissolved some of those capillaries temporarily. They've kind of eaten their way into the lining. And therefore, what it's going to look like is more like this. Come down and then go around like this. So it looks like that whole ball of cells has burrowed into the lining. It's been taken inside. So you have your capillaries running out here. Still have some. Of course, don't try to copy mine exactly. As long as you have little wiggly capillary lines there, it's good enough. Actually, I want to leave room to write. I'm going to steer that one into here. I'm going to write something right here in this space. This is an actual photograph of what we're drawing. This is the blastocyst. This would be the inner cell mass, and these outer cells. And you can see how it's kind of buried into the endometrium here. Now, it's very difficult to see the little blood vessels and things. These types of pictures are very confusing unless you know what you're looking for. All these little dots here could be blood cells, which we're not going to draw. And I'm not sure how to interpret all of this, but that just shows you why we draw and look at diagrams a lot, because the scientists know how to interpret this, and then they make it easier to see by making a diagram. But that's an actual blastocyst in a uterine wall. Now, I don't know if this is human. It could be a mouse or something, but it would look the same regardless, no matter what mammal. It would pretty much look like this. Okay, but before I color it, I'm going to do some labeling. Actually, I'm going to extend these cells. These might come around here a little bit too. And there's a special word for this outer ball now. This outer ring becomes the chorion, C-H-O-R-I-O-N. It's a super important word, the chorion. So we call these cells here chorionic villi, the little finger cells belonging to the chorion. Now it kind of looks like this whole lining is pregnant, doesn't it? It's kind of funny. And you might think that, oh, look, I know what's going to happen here. This is going to become the amniotic sac, right? Here's the baby, and here's the amniotic sac. But it actually turns out to be a lot more complicated than that. This does eventually, it stays around the outside, and it does eventually, in essence, become sort of part of the sac. But it's a little more complicated than that, you'll see. So another note we want to put here before we color it is that this area right here where the mother's capillaries and the baby cells are almost touching has a special name, and I'm sure you know it already. It's called placenta. Now, this isn't a fully formed placenta yet. This is the start of the placenta. The placenta will become a lot more complicated, and we're not going to get into drawing it, but this is the beginnings of what will become the placenta. So it's important to note that the placenta doesn't belong to the mother and it doesn't belong to the baby. It's both. The placenta is a complicated structure that has a mother's side and a baby's side, kind of all woven together. So let's add one more note down here and then we'll color. The chorionic villi begin to secrete, and you need to write in this blank, HCG. And this stands for human chorionic gonadotropin. And that's a mouthful. They usually just say HCG. This is a hormone messenger molecule that will tell the mother's body that an embryo is present. Now, of course, cells don't have eyes and ears. The way they communicate is through chemicals and hormones. So the mother's body, how is it going to know that there's an embryo down here? Like, there's no nerves. The mother doesn't automatically know. The ovaries don't know there's anything going on unless there's a message sent. So these cells make HCG, and it goes into the mother's capillaries, and then it goes into the mother's bloodstream, and it ends up all over the mother's body, and it even comes out in her urine. And that is how you can do a pregnancy test. You can test urine for the presence of HCG. So the HCG's job is to go to the ovaries, so it'll float around in the bloodstream, and most cells will just ignore it. It doesn't mean anything to skin cells or to liver cells. But when it gets to the ovaries, the ovarian cells know that this message is for them, and they know to make changes that will keep these blood vessels there, because otherwise this lining is going to just shed. So the HCG tells the ovaries, please do not shed the lining because we need it. There's a little embryo growing down there. So now I'm going to fill in around here like I did with the other drawing. And if you want to skip this, you can just cut the video off and just skip on ahead to the next one. Now let's find out what's going on with this inner cell mass. You're going to see this ring. We're not going to draw the villi and everything that's going on outside of here. You're just going to see this little outer ring, and we're going to find out how this starts to develop. So first what happens is the cells in this inner mass decide to make two layers. They get organized, and they kind of go like this. Down here, they're going to be a little bit longer, like that. And then there's a layer like this. And they have names for these two layers. This one is going to be considered on top. So the way we're going to look at the embryo in the future, we're going to consider this is the top and this is the bottom. We need a word for on top, and we're going to use epi. So this thing is called the epiblast. Remember, I think I mentioned that you see the word blast a lot in embryology, other areas of science too, but especially in embryology, you see blast quite a bit. So this is called the epiblast. And then this one is called the hypo blast. And hypo just means under. A hypodermic needle, dermis is skin, so a hypodermic goes under your skin. So hypo means under, and you can make a note if you want to Make sure you remember that. It means under, and epi means on top. And of course, this is our chorion. Okay, so that wasn't too bad. It's actually not any harder than drawing the inner cell mass. So next, this little thing moves over a bit, like this. Here's our epiblast, our hypoblast. Kind of make the cells look a little rounded if you want there. Okay, so again, this is the epiblast. And the hypoblast. And there's a little layer right on the inside here. A little layer of cells grows so that we have a little space in here. We have a little cavity opening up. So we've got fluid here and fluid here now. This is like a bridge out in the middle. And this is very important. This is called the amniotic cavity. And of course, eventually this will develop into the amniotic sac. But the way it wraps around is really clever. It's a little complicated, but very clever. Okay, so we still have two layers, and this is often called the bilaminar disc, just in case you want to know that. Let's see, where should we put this here? Let's put it right down here. Bilaminar disc. Of course, bi meaning two, lamina means plate. So that's just the two plate thing, two layers. And of course, this is 3D. It's like two pancakes. We're looking at a cross section of two pancakes. These are really round. Like if you tipped it, you'd see it kind of round inside the cavity. Okay, next, the bridge is going to move over a little bit more, like this, into the middle. And we have this little lining of cells around here like this. And then on the other side, we get the same thing. We get a little layer of cells going around this side. So now we have two cavities, right? So this is still our amniotic cavity. Same thing, amniotic cavity. And this is still our bilaminar disc. I'm not going to label them again because the names are the same. And two things to note here. This has a name. This is the future Yolk Sac, Y-O-L-K, just like the yolk of an egg. So here we have another way in which we're a lot more like chickens than we thought. The future Yolk Sac, and we have a little dot forming. I'm just going to make it a dot. Of course, it's a little place with a few specialized cells, but we're going to just represent it as a dot. And this is the start of something called the notochord, N-O-T-O-C-H-O-R-D. Notochord starts. We're going to see a lot more of that notochord here in the very near future. Alrighty, then there's a big change at the end of the week here. We're going to go from 2 layers to 3 layers. So And we're also going to have another cavity open up. So this is going to be the most complicated drawing here in our line. What we want to do is make kind of an inner circle like this, and we're going to stop seeing all the little cells now. Pretty soon we're not going to be drawing all these because our scale is going to get so ridiculously small. So like this, leave a little space and make a little, like it's connected like this. Then I'm going to make this look like it's made out of cells. So this is the layer right here. When I'm drawing, it was these cells right here that we drew right here. This is kind of opened up. Look, there's a gap now between these two layers of cells. See that this gap wasn't there over here. So now we have a gap. And this would be made out of cells, I guess. And our disc in the middle is going to be 3 stripes big. I've made mine kind of thick there. It kind of wouldn't be right that thick. Let's make it a little bit skinnier, something like this. Now I'm going to show you a picture in just a minute to show you a little nicer diagram. But 3 layers. Now it's really important that we color these correctly. The middle one is always red. In every illustration you'll see, you can Google it and look for these trilaminar discs, and you will always, 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 always see the middle one red. I've never seen one that isn't red. And the one on this side, which used to be the Epiblast, is now going to be blue. It's going to have a new name. It's going to be blue. And this one is going to be yellow. And we're going to have our dot in the middle called the notochord. Okay, so let's label everything. The outside ring is still called the chorion. This little thing here is called the connecting stalk, S-T-A-L-K, stalk. That's the connecting stalk, and you can probably guess that this is the very, very start of the umbilical cord. And we still have our amniotic sac. I'm going to see if I can write it in here real small. Amniotic cavity. Call it a cavity. It's going to be a sac eventually, but and let's just call this the yolk sac. They usually just call it a sac. They don't really call it a cavity. And we have this thing. Here's the notochord, that dot. If I can just kind of arrow to it. Oh. Notochord. And this would be the chorionic cavity. Chorionic cavity. Now getting from here to here, you can probably imagine we've skipped a few steps. There's some details into how does it exactly go from 2 to 3. Well, there's some definite rearranging and moving around that happens in the middle, and we're not going to try to draw all of it. I'll show you a couple pictures so you understand the jump, but really if you know that it goes from a 2-layer disc to a 3-layer disc and has a notochord, that's pretty much the basics of what you need to know. But just in case you're curious, I'm going to show you a few pictures. Oh, let's label this first. Let's call this the 3-layer disc, or the 3-layer germ disc. Usually what they call it, the germ layers. And that doesn't mean bacteria. In this case, germ means seed. So this is like a seed will grow into an adult plant. Well, this is like the little seed that'll grow into an adult body. So this is the 3-layer germ disc. Now let's look at a few pictures. First, I want to show you one that I find confusing. I find pictures like this. Okay, we did this, and then it goes to the morula and blastula, and I guess this would be inner cell. And then it does this. It looks like it puckers in and gets this little loop in here. And it says blastula, early gastrula, and then this is the gastrula. So it looks like the process of gastrulation is that the blastocyst gets this indent in it. So go, okay, and you see pictures like this, that looks like the same thing. However, when you look up gastrulation, which is technically what this is called, going from here to here, I could put gastrulation, you see lots of pictures where it looks like this. There's no indent. And instead, you find pictures like this, where it's straight, and I can't see any indentation. So that's not this thing. This is just that 3-layer disc, and it doesn't look dented. Now it'll do this, and we'll see that in a minute. I'll let you know what that is, but that's different. Another picture I found for gastrulation looks like this. This is a very interesting picture, and we're going to look at it for a minute. But there's a little bit of an indenting going here, but that's a far cry from what's going on here. And I have spent hours and hours trying to track this down, trying to research this and find out why they would draw it like this. Maybe that's a sea urchin embryo or something. I really don't know. It's very complicated. The best that I could find was that the word gastrulation actually means the process of going from a 2-layer disc to a 3-layer disc, which actually doesn't involve turning into a C shape. Eventually the embryo is going to fold, but right now it doesn't. What's going on is that the cells are rearranging, and this picture is really the best I could find for this. Let's zoom in. So this would be the epiblast, I guess. It would have been, this would be that Hypo Blast. And what happens is that there's a little streak. We're going to draw this in the next drawing called a primitive streak. A little fold starts happening, and the cells in the middle here start doing something very different. A section of their DNA opens up and tells them to start dividing like crazy and to start moving. And they actually migrate. They move. See, it says migrating cells. The cells start pouring down here. It's like a crowd of people being let into a room, you know, and they spread out everywhere. So they changed the color. Now, cells don't have color, obviously, but they changed it from blue to red so that you could keep track of which cells were moving. So they turn red in our drawing and then move and fill all in the middle here, and that is going to be our red layer. Some of them also go down here. You see them kind of turning yellow, and they join this bottom layer. Maybe they replace them or just join them to make our yellow layer. So the way our drawing went, it was kind of like this. Right? We have the blue, and then we're going to have the red and the yellow. So this technically is what gastrulation is. It's the process of going from 2 layers to 3 layers, and it involves cells in the middle changing, doing something different, and moving and filling this all in and creating these 3 layers. Okay, so let's look at one more picture. Now this one's a little bit complicated. Don't panic. If nothing else, I'll teach you to try at least to look at scientific drawings without panicking. So here, I'm going to use the key down here. These are our cells. This would be the nucleus, and they've labeled Golgi bodies, and the green sticks are microtubules. You know what those are. And they're pointing out that there's different kind of junctions. Now, we don't just have gap junctions here. They're telling us there's tight junctions, and there's adherent junctions, and we'll meet those in a future lesson. But they're stuck together with these little joining junctions, and there's a little membrane, the basement membrane, we're going to meet in the lessons on the epithelial cells, but it dissolves and lets these cells go down here, and then they change. See how they're making them red again? The shape changes, what they're doing is going to change, and this is a fundamental change here, because these things are going to become something very different from the ones that decide to stay on top. Very different. They're going to form different body parts. So they're agreeing, all right, we're going to go down here, and you guys are going to stay up there. So let's take a more 3D view of this right here before we go on and see how this is going to change, see what happens to the notochord and things like that. Let's draw it down here in this circle. So here's our chorion, and we're not going to make all the little cells. Let's just make a line going like this. And before we get too far down here, let's make an oval right about here. This is going to be our top of our top layer. Very narrow oval. Like this. And then another line right underneath, like this, parallel to that one. And then another line. And one more. And let's go ahead and color them. The top is going to be the blue one. And I'm going to color kind of dark there and then get lighter, a little bit lighter, so it's going to look a little bit 3D-ish. And then I'm going to make the top sort of blue also, very light. And the red layer in the middle. It's really nice that all the pictures are the same. No matter what picture you look at online, you can always see these same colors. Makes it really easy to compare pictures. The bottom is yellow. And let's go ahead and remind ourselves we have this middle one is going to have that notochord thing. And actually it's going to have a little streak right there, notochord. So let's bring this down here now like that. And then you can shade a little bit to make this look like this is hollow. So it's kind of like we have these pancakes stuck in a ball. And then we've cut the ball, but not the pancakes. In the next drawing, we're going to slice the pancakes and look at the inside of the pancakes. But right here, we can see the whole pancake. All three of them. And so you want to make these little corners dark and then come out gradually lighter and lighter. And then underneath the yellow one, we kind of want to make that a little bit darker too, because it's going to kind of shadow the underside there. And then on the top here, we can make this a little bit more shadowed up there also. I'm going to go ahead and make this all dark. I might even just bring that out a little bit. There. Alright. Now we're going to find out the correct names for these layers and what's going to happen to them. The top layer, the blue one, I'm going to point to that, and it's going to be our number 1. And that is called the Ectoderm. It used to be called the Epiblast, but now we call it the Ectoderm. Ecto means outside. Derm kind of means skin, so actually this is very descriptive. The ectoderm becomes your outside skin. Part of it does. And let's see, you need to write very small on this line. Write very small. It will become skin, and in particular epidermis. That's the outside layer. Skin is more complicated than just one layer. This is the outside layer. And then also hair and nails, things associated with the outside layer. And very importantly, it becomes the brain and all the nerves, nervous system. Skin, hair, nails, brain, and nervous system, all from that blue layer. So the next layer, the red one, is called the Mesoderm, or I've heard it called Mesoderm. I generally say meso, but you can say either one. And M-E-S-O, it just means middle. It just means middle layer. So the Mesoderm is going to become, right small here, real small. We actually have 2 lines. This is going to become a lot of things. So it's going to become muscles. Remember, put that C in there. It looks like muscles. Muscles. Bones. Connective tissues, and this would also include blood. We're going to see that blood is actually classified as a connective tissue, so it would be blood. And continuing on here, it's going to become kidneys and also heart. And it's going to become the, I'll say bladder, that means urinary bladder, and gonads. Now gonads is a generic word that means male or female reproductive organs. This would be ovaries and testes. And it was also going to become all the lymph nodes, and therefore the spleen would be part of that. And if that weren't enough, it's going to become the dermis of skin. Remember, skin has several layers, and this is going to become the bottom layer of the skin as well. So that mesoderm is really multitasking there. The bottom layer is called the endoderm. And endo means inside, so you can probably guess it's going to be the really, really inside stuff. This is going to become the lining, the inner layer of, and I'm just going to put gut because it's a short word, and we know it means a whole digestive system. So the lining of the gut, oh, and lungs, we can add, it's the lining of the gut and lungs, the inside of your lungs. also becomes your liver, your pancreas, and lots of glands. We'll put glands, thyroid, thymus, lots of glands, lots of glands. Now, why does any of this matter? I mean, these are going to turn into other things and kind of disappear, but scientists like to keep track of where things came from, these 3 layers. And one thing that they use this for is to classify types of cancer. And you can see my note here about after all the body parts are fully developed, the cells are programmed to slow down and stop dividing. We're going to get all these body parts made, and then we don't need 3 livers, we need just 1. They have to stop. But rogue cells are cells that didn't get the message, and they decided to go right on dividing and dividing and dividing, and you don't need a liver the size of a pickup truck. The size you have is fine. So they continue to divide, and then they cause tumors and different types of cancers. Cancer is just too much of a good thing. The cells just don't know when to stop dividing. Dividing is not a bad thing, but when you do it too much, it becomes a bad thing. So some of these can cause childhood cancers. So what if some of these parts just kept going and going and going and going? And in fact, cancers that arise from the mesoderm are called sarcomas, S-A-R-C-O-M-A. I'll make it plural, sarcomas. Cancers that arise from the mesoderm are called sarcomas, and cancers that arise from the ectoderm or the endoderm are called carcinomas, C-A-R-C-I-N, carcin-O-M-A, and I'll put an S to make it plural so grammar's correct. Cancers that arise from ectoderm and endoderm are called carcinomas. And interestingly, this type of cancer from the ectoderm and endoderm are more common in adults. And the sarcomas are more likely to be found in children and teenagers. So I don't really want to end on a sad note, but that's just something interesting about these germ layers where you do see it in other branches of science. So it's just something that's interesting to know. And now we're going to look at how these layers develop into these things.